So nice to meet everyone. I'll get started while the slides are coming up, and sorry for the abridged timeline here, but my name is Rajeev Malhotra. I'm a cardiologist and critical care. Oh, that's okay. Can you hear me now, or is that okay? Okay, I'm Raj Malhotra. I'm a cardiologist and critical care doctor at Mass General Hospital, but I take care of adults, so that means that my patients are not as cute as Patty and Mark's patients, for sure. But in addition to taking care of patients, I have a vascular biology laboratory, and so we're very interested in the biology of vascular diseases, particularly related to the smooth muscle cells. And I think the common theme today is, and goes along with what Dr. Milovich was saying earlier, is that you can learn a lot about adult vascular diseases from pediatric diseases and vice versa. And there is a lot of overlapping mechanisms between the two. Let's see, can I forward with this or this slides?
Yep.
Okay, there we go. So I'll chat. So it's a real pleasure to meet all of you, and I think we all have a common goal, which is to get therapies available for this devastating disorder. And of course, when we think about MSMDS, we know it's a rare genetic disorder that's caused by mutations in the ACTA2 gene, which is a smooth muscle disease gene. And in that setting, all the organs that are commonly, known to have smooth muscle cells are affected, like the eyes, the heart and the aortas, the blood vessels. The gallbladder doesn't contract as well. The lung has smooth muscle cells in the airways that are affected and can cause asthma and lung disease, the intestines and the bladder. But interesting, what we're going to talk about today is that, is there an effect on the lymphatic system and the immunologic system? And it has been established, in small numbers that patients with MSMDS do have a higher rate of upper respiratory infections, as well as a longer recovery time from these infections. And so you could attribute that potentially to the smooth muscles in the airways and the inability to clear the infections. But one question that has come up is, well, is there an actual effect of ACTA2 on the immune system directly? And so with the animal model that was developed by Mark and Patty, we can actually interrogate that question. And so at about the time that I started working with Patty and Mark on this project of rare vascular diseases, we were very fortunate to recruit Dr. Kangsan Roh as a faculty scientist in the laboratory, who happened to also be an expert in lymphatic diseases. So sometimes the best discoveries occur when you intersect two different areas or specialties. And so our lab, having focused on the vasculature and his prior experience on the lymphatics, set us up for that. And so he had previously, prior to coming to the lab, had done this interesting experiment in mice where he isolated both the arteries, the carotid artery and the femoral artery, as well as a long lymphatic vessel, and harvested them in a way that you could isolate them at the single cell level, and then performed high-resolution sequencing analysis on each individual cell. And this allowed us to do what's known as a single-cell analysis, where at each individual cell, we can look at the differences in their phenotypes. And of course, when you isolate a blood vessel, I think we all know that the blood vessel consists of endothelial cells, smooth muscle cells, fibroblasts, immune cells. So we can see all of those here, in different colors. And then the lymphatic vessel actually has a similar anatomy. You have lymphatic endothelial cells, you have lymphatic muscle cells. And so when we take the lymphatic muscle cells and compare them to the smooth muscle cells in the arteries and put them on the same plot, they actually look diametrically opposed. They're very different, so they express a lot of different genes. But at the same time, the contractile apparatus, because both are responsible for the contraction of their respective vessels, the contractile proteins are the same. And so they both have a very high degree of expression of ACTA2. And so that led us down this road of questioning, well, okay, so if ACTA2 is highly expressed in the lymphatics, do patients who have dysfunction of ACTA2 exhibit abnormalities in the lymphatic system? And so Kangsan's going to be presenting his data that he developed in the laboratory over this past year to answer this very question. I'm just going to go over an overview of what the lymphatic system is. And so when you think about the circulation, there's the arterial system carrying oxygen in a high pressure flow system to all the different parts of the body through the aorta. And that oxygen then gets transferred to the tissue in the capillaries, the smallest vessels in the body. And then in the capillaries, also carbon dioxide and toxins are brought into the blood for secretion through the venous system and out through the lungs. The carbon dioxide is breathed out through the lungs. But in this high pressure system, and certainly in different disease states, the arteries can become leaky. And so even under normal situations, through this high pressure system in the arteries, the hydrostatic pressure pushes fluid out into the extravascular space, or what we call the interstitial space here. And that causes the secretion of interstitial, or the development of interstitial fluid. And so that's where the lymphatics come in, because the lymphatics, which have a parallel circuitry to the veins and the arteries, they pick up this extra interstitial fluid and return it back to the circulation. But along the way, when it picks up this fluid, that fluid, first of all, similar to veins, these lymphatics have valves that force the fluid in a one-way direction. But also along the way, you make stops in these lymphoid organs or immune organs called the lymph nodes. Okay, and so in addition to picking up interstitial fluid, this lymphatic tree also picks up immune cells and can reduce inflammation in different tissues and organs. And so when we think about all of the different functions of the lymphatic system, it's not only to return this 10% of extra fluid back into the circulation, but it also transports nutrients, transports lipids and cholesterols, is important for tissue regeneration, but also importantly transports immune cells, which is critical for the defense against infections. And so, so what if something wrong happens to the lymphatic system? So I know not the prettiest sight here, but these are diseases that we do take care of in our adult populations. But lymphatic diseases affect about 6 million patients in the United States. And most of it's due to something called secondary lymphedema. The vast majority have secondary lymphedema, although patients can be born with lymphatic malformations, or they can develop an overgrowth of lymphatics that can cause dysfunction of the lymphatic system. But basically what ends up happening is that when you have a dysfunction in the lymphatics, you can develop significant swelling in your legs. And that's because you're no longer able to return this excess interstitial fluid back into the circulation. And this can result in significant compromise of mobility or blood flow to the lower extremities. And unfortunately, there's absolutely no therapy to date for lymphedema or any abnormalities of the lymphatic diseases. Lymphatic dysfunction is also associated with different metabolic disorders like obesity, cardiovascular diseases, cancer, as well as brain diseases, where we know that in dementia, age-related dementia, abnormalities in lymphatics have been found to reduce the ability to remove waste products from the brain, such as amyloid deposition. And so we became, through this, random chance of studying ACTA2 in the lymphatics, we became interested in trying to develop novel models and therapies for lymphatic disease. In addition to the circulation, as I mentioned, the lymphatic system regulates the immune system. And so as the lymph fluid travels back up into the circulation, it will make stops along the way in these immunologic organs called the lymph node. And that's where B cells, T cells, important immune cells are housed and can fight our infections. And so the process of fighting an infection involves, well, first, something foreign has to be attacking our body, whether it be a virus or a bacteria. And that foreign material is picked up by certain immune cells. Let's call them macrophages, although there are other cells as well. These macrophages start detecting this foreign material, foreign virus or bugs. And then once it takes them in, parts of these proteins of these foreigners, it will then move and migrate through the lymphatic system to the lymph nodes. It will then present these antigens or foreign structures to the B cells and T cells in the lymph nodes, and that will cause those B cells and T cells to start proliferating, the ones that are specific that enable us to fight that specific virus or that specific bacteria. And that's actually how vaccines work. You give sort of this foreign agent to your body, like the COVID vaccine, and these macrophages pick them up, take them to the lymph nodes, and then cause the immune cells in your body to the ones that recognize those specific foreign structures to proliferate and then attack the foreign bug. So the lymph system clearly plays an important role in fighting infections. Interestingly, the lymph system has effects on organs that you wouldn't think you would have an effect on, organs that don't typically get infected. And so I'm just going to put the example of the heart here, which is sort of near and dear to what I study. But just imagine a patient having a heart attack, right? So part of your tissue now gets damaged from the blockage of blood flow to the heart. In that context, when you damage that tissue, you no longer are able, the lymphatic structures also are damaged. So there are lymph vessels that traverse the entire heart myocardium. And so when you damage the lymphatic structure, unfortunately, that means that fluid can no longer get into the lymphatic structures and you can't uptake immune cells. So what does that mean to the damaged part of the heart? Well, there's a lot of remaining fluid or swelling that remains, and that's called edema. And the immune cells that are there are trapped there, and they end up laying down a lot of collagen and fibrosis and scar tissue, and that causes the tissue to die. So even in infected states, the lymphatics play an important role in regulating inflammation of the tissue and swelling or edema in the tissue. And so right now, therapeutics are being currently developed in our lab and other labs to try to restore this ability of lymphatics to grow in tissues that are fibrotic. So what does a lymph vessel look like? It actually looks similar to sort of in between an artery and a vein in some ways. So you have these structures that are lined with lymphatic endothelial cells in red. They have contractile apparatuses to move the fluid in the right direction with these lymphatic muscle cells or lymphatic smooth muscle cells. And they also have valves that prevent the fluid from going in the wrong direction. They're sort of one-way valves like veins have. And so in that setting, we can study using some of the same tools in the laboratory, the same way we study arteries, we can study the lymphatics. And so, Kangsan is an expert at doing these tools, and so you'll play the video on the right, but this is the stain of ACTA2 right here in red. There's a ton of ACTA2 expressed in this lymphatic vessel here. And you can actually see, using ultraviolet microscopy, the actual contraction and relaxation of these lymphatic vessels. So using these cool tools that we have in the laboratory, the questions that we had are: one, what is the role of ACTA2 in the lymphatic system? Two, using the mouse model, are there abnormalities to the lymphatic system and the immune system with ACTA2 mutations? And three, does this correlate to any of the phenotypes that we see in patients with MSMDS? So I'll turn it over to Kangsan to present his data.
And sorry about that. I'll play it back in.
You're good?
Thank you for introducing me, Raj. Hello, I'm Kangsan Roh. I'm instructor of the Cardiovascular Research Center at MGH. It's so nice to meet you. I've studied lymphatic biology with a focus on the lymphatic diseases causing dysfunctional lymphatic infections. I've been studied on the role of the lymphatic vessel in the multisystemic smooth muscle dysfunction syndromes. One of the biggest challenges in challenges in studying MSMDS has been the lack of the appropriate animal model system. But thanks to the Patty and Mark, the MIH11 active flux mouse was created. So as a MSMDS mouse model, so we can overcome this limitation. So it offers a very several key advantages. This mouse model carries the ACTA2 or arginine 179 histidine mutation, which has been identified in human MSMDS patient, and is known to be one of the most pathogenic variant with the disease. This mouse model is knock-in mouse model, so the cell expressing the Cree recombinase will be knock into the arginine 179 mutation in native ACTA2 gene. So this setup is very powerful because the MIH11 promoter is only active in the vascular small cell. So the MIH11 CRE is expressed in lymphatic muscle cell as well. So that means the mutation is turned on only in the vascular tissue. So it allows us to study tissue-specific disease mechanism without affecting other parts of the body. So these mutant mice has a lower survival rate and reduced body weight. And so they have very significant the phenotype compared to the control mice. So in the active mutant mice, especially the abdominal organ, the small and large intestine, so they are very significantly distant, resulting in the overall abnormal abdominal swelling. And this suggests the GI, the gastrointestinal dysmotility. Additionally, the ACTA2 mutant kidney showed the high hydronephrosis. This can be the bladder is also observed, suggesting urinary retention or the impaired the bladder emptying. So we've been using this mouse model to study the underlying mechanism of the MSMDS and to investigate how the ACTA2 work affect the tissue in the lymphatic system. So here's the rationale behind our study. First of all, the patient and the mouse model both showed very clear signs of the vascular small cell dysfunction that already established for now. But when it comes to the lymphatic system, we don't really know much about it. Even though these patients often have some problem with their immune responses and the swelling issue, but nobody has looked into the MSM that affect lymphatic function specifically. So what's interesting is that the ACTA2 is highly expressed in the lymphatic muscle cell, which are on the responsible for the plumbing, pushing the lymph fluid forward through their spontaneous contraction. So based on that, we hypothesize that ACTA2 mutant mice might also show signs of the lymphatic and immune dysfunction. To evaluate the structural changes in the collecting lymphatic vessel of the ACTA2 mutant mice. So we measured the coverage of the lymphatic muscle cell and the lymphatic diameter in the peripheral collecting lymphatic vessels of the popliteal tissue. We found that the coverage of the alpha-smooth muscle actin positive lymphatic muscle cell was significantly reduced in ACTA2 mutant mice. In the same collecting lymphatic vessel, we found that the podoplanin is the marker for the lymphatic endothelial cell of the collecting lymphatic vessel. That endothelial cell layer in the same collecting lymphatic vessel appeared very dilated. So that indicates the structural impairment of the lymphatic vessel. For this part, we measured the lymph flow, lymph drainage through the draining lymph node assay. Basically, we injected the fluorescent dye into the poop pad and intradermally, and then tracked how the dye moved through the lymphatic system over time. For at the 40 minutes after injection, we saw in the control mice, the dye had already get to the inguinal lymph node, but in the active mutant mice, the dye didn't make it that far. Then by 1.5 hours post injection, the dye in the control group had reached all the way up to the axillary lymph node, but in the contrast, the mutant mice, they still showed impaired flow, and the dye didn't reach the axillary lymph node. So to evaluate the contractile function of the lymphatic vessel in the ACTA2 mutant mice, we performed the intravital lymphatic imaging. So can you play the both video, please? So we injected the fluorescent dye into the poop pad, and we could observe that the dye the flowing into the populated collecting lymphatic vessel like this. And to measure the lymphatic pumping, we capture the 360 frame over 80 milliseconds. So yeah, to the left one, please. Yeah, I think the video is not working for now. So we measured the 360 frame in 80 milliseconds using the inverted fluorescent microscope. Then we quantified their change of the diameter, and we allows the evaluate the contractility and their frequency. So in the control mice, it showed the very dynamic pumping in the and it showed very clear and dynamic contraction and very higher the frequency. But in case of the active mutant mice, it has very lower contractility and their frequency. So ectotic mutant mice was their lymphatic contractile function was significantly reduced. So since because the MSMDS is a disease that caused the multi-organ smooth muscle function, so we wanted to see how the ectotic mutation affect the lymphatic structure all across different organs, so including the brain, aorta, heart, and the small intestine. So actually we checked the more different types of the tissue, the liver, the bladder, and so on. But I brought this only for the major tissue. So we looked at when you looked at this brain, the meningeal lymphatic vessel in the brain, the aortic lymphatic vessel in the vein of the aorta, and the cardiac lymphatic vessel in the heart epicardium, and the lacteal lymphatic vessel in the small intestine and villi. So in all of these reasons, we observed the dilation of the lymphatic vessel in the active mutant mice, which suggests that the widespread structural abnormality that caused by the mutation. At our institution at MGH, and as part of the collaborative effort with the MGH group, we have access to the very valuable plasma samples from the 12 MSMDS patient and age-matched health control. Using the Olink platform, we performed the proteomic profiling to compare the protein expression patterns between these two groups. The results show very striking the differences in protein expression, the profiling between the groups. Specifically, we found that the many factors that related to the immune responses and immune regulation and lymphoid organ development were significantly reduced in the MSMDS group. This means MSMDS might involve more the immune system and the lymphatic problems than we currently understand. Among these, the significantly differentially expressed protein, we focused on the lymphotoxin beta receptor, we call it LTB beta R, and the TNFS SF13B, called as B cell activating factor, both of which are very essential for the lymphoid organ development. So these proteins are notably downregulated in MSMDS patients. The lymphotoxin beta receptor signaling is very essential for the formation and the organization of the secondary lymphoid organ, like lymph node and spleen, like that. And particularly in the establishing germinal center microarchitecture. The germinal center help B cell mature and improving antibody strength and diversity, and generating long-lasting immune memory. In mice, the lymphotoxin beta receptor deficiency then result in the failure to form the germinal center. Likewise, the BAFF is a key regulator of the B cell maturation and germinal center responses. So the BAFF support the B cell survival, and in its absence, the germinal center B cell failed to develop properly. So in this paper, so the mouse model has shown the BAF deficiency significantly impaired the germinal center formation and T follicular helper cell development and antibody production. Based on these findings, we hypothesized that the immunological abnormalities observed in the MSMDS may arise from the defective germinal center development in the secondary lymph node organ, potentially linked to the ACTA2 mutation driven dysregulation in this mouse model. So here we are looking at how the ACTA2 mutation affect the immune microenvironment and lymph node function. Firstly, you can see here we collected the mesenteric lymph node in the mesenteric area, and when you compare them between the control group and mutant mice, you can see the image on the left. The mesenteric lymph node from the ACTA2 mutant mice are visibly smaller than those of the control group. To get the better understanding of the immune cell population, so we performed the flow cytometry analysis on single cell suspension from the mesenteric lymph node. So GL7 positive, GL7 B220 double positive germinal center B cells are significantly reduced in the active mutant mice group, and this reduction was also confirmed by the immunohistochemistry of the GL7 staining, which mark the germinal center B cells. It shows a very strong signal in the control group, and they have very well organized B220 positive B cell follicle. But in case of the mutant mice, the GL7 signals looks very scattered, and they're not the center, and their morphology was not stable. So altogether, this finding suggests that ACTA2 mutation lead to the altered B cell immunity and this dysfunctional lymph nodes, the architecture. So this result to show the mesenteric lymphatic drainage and lipid absorption are disrupted in the ACTA2 mutant mice. So to evaluate the mesenteric lymphatic function, we used the butterfly C16, that's which is a fluorescent lipid tracer. So after the 24-hour fasting, and then the tracer was treated through the oral garbage, and this fluorescent imaging was performed after 2 hours. So in the control mice, the tracer was efficiently observed through the mesenteric collecting lymphatic vessel, and we can see that the mesenteric lymph node has the signal of this lymph flow from the small intestine. But in case of the ACTA2 mutant mice, we cannot see any signal through the mesenteric collecting lymphatic vessel, and we can see very few or non-signal, or almost non-signal of the butterfly C16, the mesenteric lymph node in ACTA2 mutant mice. So this suggests impaired the lipid transport and the defective lymphatic absorption in the ACTA2 mutation. Additionally, immunofluorescence staining of the mesenteric collecting lymphatic vessel showed abnormal vessel dilation and the marked reduction of the alpha SMA positive, the lymphatic muscle coverage in the mutant mice compared to control group. To further explore the immune dysregulation in the ACTA2 mutation, so we looked at the CCL21 as a very popular key chemokine involved in the lymphatic homing. So as you can see here, so the green color, the CCL21 expression is significantly reduced in the live one, the mesenteric lymphatic vessel of the mutant mice, because CCL21 is very critical for the guiding the dendritic cells and naive B cell and T cell into the lymph node. for the antigen presentation and presentation. So its downregulation likely contribute to the poor immune surveillance and the defective adaptive immune responses in ectotomy mutation mouse. So in short, this result add another layer of the evidence that lymphatic structural abnormalities in the ectotomy mutants are directly linked to the functional immune impairment. For the gene therapy, specifically targeting the vascular smooth cell, we are collaborating with Ben and Casey, Mark and Patty on this. So we are using the dual AAV the system here because the full base editor and the Cas9 are too big to fit into the one vector. So this introduction and explanation of the AVPR is already done very much as I know. So we tested the MSMDS mouse model. We are able to get the around 10 to 50% of editing correction. So the most importantly, we saw the very clear improvement of in the survival and the treated group compared to the untreated group. So this collaboration allow us to show the precise in vivo correction of the ecta2 mutation is possible and has a real therapeutic impact. So to find out whether these therapeutic effective, the AVPR can actually reach the lymphatic vessel and lymph node. So we use a TD tomato reporter mice. So we intradermally injected the AVPR CBA3 vector into the poop pad, the 3-day-old pup, and then we observed that we dissect them at six weeks. Then we harvested the draining lymph nodes and the lymphatic vessel from the poop pad area. So when we imaged the tissue, checked the AVPR transduction, we found something interesting. Very strong AVPR vector signals detected all lymph nodes, and we also confirmed the mesenteric vessels were successfully transduced. So this is TD tomato reporter mice. So I stained the lymphatic muscle cell with the alpha SMA, and the blue dot is the prox one is for the lymphatic endocellular cell specific marker. So this yellow color indicates that the AVPR vector is co-localized in the alpha muscle actin positive lymphatic muscle cell. So in conclusion, our study provides new insights into how the ecta2 mutation associated with the MSMDS impact the lymphatic and the immune system. So the arginine 179 mutation in ecta2 gene lead a very significant loss of the lymphatic muscle cell coverage and contributing the development of the lymphatic aneurysm. And this mutation caused the widespread dilation of the lymphatic vessel across the multiple organ system, highlighting the systemic defect in the lymphatic structure. As a result, the lymph flow was impaired, and the contractile function of the collecting lymphatic vessel is significantly reduced. Then the proteomic analysis of the plasma from the MSM patient showed distinct the molecular signature associated with the immune dysregulation and disease progression. In mouse model, in ecta2 mutation altered the immune microenvironment within the secondary lymph node organ, suggesting that this disrupted the immune lymphatic crosstalk. Finally, we demonstrate the successful delivery of the proof of concept of the AVPR gene therapy vector to lymphatic vessel and lymph node. Our ongoing work is evaluating whether this approach can restore the lymphatic structure and the immune remodeling in the ecta2 mutation mice. The further study are needed to evaluate the long-term therapeutic potential of the AVPR gene therapy in MSMDS mice, including the survival rate and the lymphatic function and immune restoration. And I plan to characterize the immune cell population of MAC2 mutant mice, including the PBMC and bone marrow cell, to find out their progenitor cell remodeling. Yeah, sorry. That concludes my presentation. I would like to mention my mentor Raj and all my collaborators who have contributed to this project, and I very sincerely hope that these findings and further the discovery will contribute to the finding any clue to find a better therapy for the MSMDS patient. Thank you.
Good presentation. Thank you very much. Do we have any questions?
I'll just congratulate Kangsan on such beautiful work, and I think it's really threefold discovery. One, defining the role of Acta2, not only in the blood vessels, but in the lymphatic circulation and the immune system. Two, we now have a new mouse model, the Acta2 mutant mice, that is a model of lymphedema. And three, I think this is the first proof of concept presentation of a gene therapy that can actually target the lymphatics. So it could potentially hold promise for development of therapeutics for those 6 million people with lymphatic disease.
Thank you very much.
Thanks so much, Frank.
Thank you.